plants. Now, before we talk about the actual process of transportation, and before we start discussing the different structures in the plants, we first need to understand why do we actually need a special transport system in the plants. Why do we actually need a special support system? What's the need, right? Why is there a need of a special transport system? That's the main question. Now, there are a lot of different reasons why we require a special transport system in plants. The first one is pretty obvious. We need that transport system to move substances. Now, there is one thing that we have to consider. In plants, right? So I need to move substances from the site of absorption. For example, the roots, right? The roots are the site where are actually absorbing all the nutrients, all the minerals, all the required nutrition, right? To where they are needed. So that would be the growing plants of parts of the plant, your shoots, right? Your shoots, your leaves, your fruits, your flowers, all of these require nutrition. So I need to move substances from the area of absorption to the area where those nutrients are actually required. Then I also need to move substances from where they are produced. That would be your leaves, right? Leaves are the primary site where you actually make your glucose. So move substances from where they are produced to where they are needed for metabolism. Again, this when you're going to go ahead and talk about it, this is going to include what? This is going to again include the growing parts of the plant. So whatever the growing parts of the plants are, this could even be the roots. If the roots need energy to grow, your roots would also require some sort of nutrition. Then I also need to move substances to different parts of the plant for storage, right? Because it's not just about making the food and absorbing the food and metabolism. I also need to store the glucose that is being made. For example, a good example would this be your potato plant, right? Potato plant, as you know, the potato plant, the potatoes are tubers that can be found under the soil. And what happens that the energy is stored or the sugar is stored or the glucose is stored in that potato plant for whenever it may be needed, right? So that is another primary reason why we need to have a proper transport system to allow all of these movements to take place. Now, so when we're going to talk about it, what's then, oh, fine. So we require a transport system. Okay. Now, question when you're going to talk about it, then the question comes, fine. What does the transport system include, or what is the transport system? We understand why we need a transport system, fine. But okay, what is this transport system that we are studying? Now, the name of the transport system and a few basic points you guys have already studied in your IGCSE. The transport system, there are two transport systems, as you guys know. We have our xylem vessels. Xylem vessels, as you know already, are for transport of water and mineral ions. Keep this in mind that both mineral ions and water are transported in xylem vessels. And then, of course, we have our phloem vessels. Phloem vessels are going to be involved in transport of sucrose, not glucose. We don't transport glucose as it is. We convert glucose into sucrose and then we transport it into through the entire plant. So glucose is converted to sucrose and then it is going to be transferred through the phloem vessels. The term we use for it is we call it phloem sac. Sap is basically the fluid, the sucrose containing fluid that you can find within the phloem vessel. And when we're talking about the xylem vessel, so the xylem vessel carries the water from the roots to the other areas of the plant. The movement is unidirectional. Unidirectional means that it only takes place in one direction. The movement is in one direction only. It's from the roots to other parts of the plants. When we talk about this, our flowing system, so our flowing system, right, is multidirectional. What do I mean by multidirectional? What I mean by multidirectional is that at one time, phloem vessels can transport sucrose in different directions. So you can have, for example, that sucrose can go from the leaves to the flower, from the leaves to the shoot, from the leaves to the roots, from the leaves to the fruits, right? So this is an important difference between xylem vessels and phloem vessels. Xylem vessels are unidirectional, meaning that the water will only go in one direction, that is away from the roots. In phloem vessels, the sucrose is carried away from the leaves. It can be carried towards the leaf. It can be carried towards the root. It can be carried anywhere at all, right? So that is an important difference that is present in both your transport system, xylem system, and your phloem system. Now, you guys need to know in detail the structure of both xylem vessels and phloem vessels. Okay, in detail, you guys need to know the xylem vessel and phloem vessel, the difference in your phloem vessels and in your xylem vessels, right? It's really important. Now, before you and I go over the structure of what a xylem vessel looks like and what a phloem vessel looks like, we actually need to go over the structure of your stem, the structure of roots, and what are the different parts of these structures. Okay, so we have to go over that first. We first need to go over the structure of leaves, roots, and your shoot, your stem, and then we can take a look at the special structure we see in xylem vessels and phloem vessels. Now, generally speaking, let's do a small revision of what you guys had covered in IGCSE. If you guys remember, you had studied about two types of flowering plants, monocotyledons and dicotyledons. Monocotyledon only had one cotyledon inside the seed. Dicotyledon had two cotyledons inside the seed. And you guys might have studied the differences between monocotyledons and dicotyledons. For example, if you remember, monocotyledons would have long, narrow leaves, right? Dicotyledons would have, broader leaves and they would have stalks attached to the leaves, right? So, the what you and I in your syllabus, you're only supposed to know the structure of dicotyledons, not monocotyledons. Okay, keep that in mind. So let's go ahead and start by looking at the structure of our stem. Okay, let's start by looking at the structure of the stem. So when I'm talking about the structure of the stem, what am I going to see? Give me a second. Let me actually first draw it out for you guys. Tell you guys the labeling. Now, you guys are supposed to know the structures at a low magnification and also at a higher magnification. Okay? First, let's talk about what we will see at a lower magnification. I will show you guys a proper slide as well, like taken from a microscope. I'll show you guys the slide as well. And I'm going to draw the plan diagram for you guys as well. And if you guys get in the exam, what does the plan diagram actually look like? Hold on, let me just draw this out one second. All right, so this what I've drawn for you guys. This is basically the structure of your stem. Okay, and specifically speaking, this is a low power or a low magnification, I should say. Low magnification drawing of the these are the structures you will see at low magnification. All right. Now, if I go ahead and I talk about the labeling, so this outermost layer is known as epidermis. All right. Now, over here, this in the middle, this is going to be known as what? This is your phloem. Okay. Now, when you're making a plan diagram, you don't need to show the individual phloem vessels. All right. You don't need to show the individual phloem vessels. But just for the sake of explanation, I will draw them. Now, oftentimes you guys will see your past paper questions in your paper 2, and they will give you, like pictures of the light microscopy. And they will ask you to identify which is the xylem vessel and which is the phloem. Now, keep one thing in mind that your xylem vessels are generally larger in size compared to phloem vessels. Okay, so you will see something like this. You will see that the xylem vessels will be larger than the phloem vessels. Okay. Now, again, you also have to remember the order. Phloem vessels in the stem, right? Root might have a different situation. In the stem, the phloem vessels are on the outside, while the xylem vessels are on the inside towards the center of the stem. Okay. Now, over here, when you're going to go ahead and talk about it, we were practicing our labeling. Now, what is this structure that is surrounding the xylem vessels? The structure that is surrounding the xylem vessels, they are your fibers, right? What type of fibers they are, that I'm going to tell you guys in just a little bit. I will tell you guys what kind of fibers they are. We will discuss them. But these are your fibers. When we are going to do high magnification, high detail, then I will tell you guys the different types of flowers that we are going to see. So anyway, this is the structure. This is the, you have fibers surrounding what? Surrounding your phloem. And then of course you have your xylem vessels. Now, between the phloem vessels and the xylem vessels, we have a layer of cells. This layer of cells is known as cambium. So this is of the stem. Please keep this in mind. Right now, we will also do the structure of leaf and the structure of roots. So they all have different structures. All right, you guys need to make sure you keep that in mind. This is the structure of your leaves. Okay. So when you're going to go ahead and talk about it, this is what the structure of your leaf is actually going to look like. So when we are going to go ahead and talk about it, we also need to then go ahead and take a look at the, okay, yes. And all combined, if you want to combine everything, what will we call it? We will call it vascular bundle. All combined, this will be known as what? Vascular bundle. So this is what the structure of your stem is actually going to be looking like. Now, let me show you guys the actual image, like from a light microscope. What are you guys going to see? Hold on, give me a second. I'll just share with you one second. See, this what you see in front of you. This is your low powered or low magnification view of the stem that you will see from the light microscope. So this over here, this is or this particular region you see in the center, this blue region in the center are your phloem vessels. Then over here you have this sort of purple layer in the middle. This purple layer is known as your cambium. Cambium is basically unspecialized cells that will differentiate to form new phloem vessels and new xylem vessels. Okay, so it's cambium is basically unspecialized cells that will differentiate to produce new phloem vessels and new xylem vessels. Now over here, can you see these large vessels? These large vessels are all xylem vessels. All right, and these sort of purple cells that you can see, these purple cells that are surrounding the vascular bundle, these are your fibers. And I'll tell you guys the name of these fibers later on. Okay. This over here, the outermost layer of cells is your epidermis. Okay. And the cells you see in the middle, not as a part of the vascular bundle. This is one vascular bundle. The cells you see in the middle, not a part of the vascular bundle, but the cells that are just lying in the middle, they are known as your cortex. All right. So this is what it actually looks like. This is what it looks like. So they will actually in your paper too, just show you a picture like this, give you a picture of the light microscopy, and then ask you guys to label the different parts. And then they will ask you different functions related to it. So you guys need to know the labeling of your structures. So just to go over it once more, this outermost layer is your epidermis, okay? Then this is your vascular bundle. All of these are your vascular bundles. Then in your vascular bundle, the middle region over here is your phloem vessels. The large vessels in the center are your xylem vessels. The layer of cells in the middle is your cambium. Cambium are unspecialized cells that will differentiate to give you new xylem vessels and new phloem vessels. And then you have these cells that are in the middle of the or in the between the vascular bundle. The cells that you see in the middle of the vascular bundles are your cortex. So this is the structure of your stem. Now, similarly, we need to go ahead and take a look at the structure of our root. And then we actually need to take a look at the structure of our when you're going to talk about it, the structure of our leaf. Okay. When I'm saying structure of leaf, so we're actually going to be taking a look at a transverse section, which I will show you guys in just a minute. But before we actually do that, let's go ahead and take a look at what the structure of a stem. Sorry, I've already told you guys the structure of a stem, what the structure of a root is like. Give me a second. I'll just draw it out for you guys. Hold on. This is showing you a low magnification structure of your root. Now, this outermost layer that you can see, this is known as epidermis, just like your stem. In the stem, it was known as epidermis. Similarly, over here, it's going to be known as the epidermis as well. Now, when you're going to go ahead and talk about it, inside the epidermis, you're going to have your cortex. Then, inside the cortex, the vascular bundle is towards the center of the root. Remember, this was a difference we studied in IGCSE as well. That in stem, the vascular bundles are arranged towards the outside, while in the root, the vascular bundles arranged towards the middle. So this outermost layer is known as endodermis, which I will teach you guys about in just a minute. This outermost layer is known as endodermis. Then inside the endodermis, we have another layer. That layer is known as pericycle. Again, I will teach you guys about what pericycle actually is. I'll tell you guys what it is. Right? Now, inside the pericycle, we are going to have our vascular bundle. Now, in the vascular bundle, the arrangement is somewhat like this. This that you see on the outside, these cells that you will see on the outside, these are the phloem cells. So phloem cells will be on the outside. And then these cells that you will see in the very center, these are your xylem vessel cells. And this is what the structure is going to look like when we're going to talk about our root. Now, let's go ahead and take a look at what the structure is going to look like when we talk about leaf. Okay. So when we're going to talk about leaf, some of the structure that we're going to go over is going to be somewhat similar to what you guys have done in IGCSE. But a lot of the structure that we're going to take a look at is going to be new for you guys. Okay. So when we're talking about the structure of leaf, it's going to look somewhat like this. So I'm going to have my leaf. I have cut the leaf in the middle. Like I've cut like if this is my leaf, like this, then I've cut it like this. Okay, this is the section we are seeing. I've cut the leaf like this through the middle. Horizontally, not vertically, horizontally, not longitudinally, horizontally. Okay. So the leaf is going to look something like this. This is my leaf. Now, when we are going to go ahead and we are going to talk about it, first of all, we're going to have the outermost layer. Okay. So the outermost layer that's going to be present at the top of the leaf, and then we will have an outermost layer that's going to be present at the bottom of the leaf. Now, when we're going to talk about it, if you guys remember, we used to have an outer layer that used to exist on top of the leaf. If you guys remember the name of it, so it was known as the cuticle. What was the function of the cuticle? The function of the cuticle was that it was a thick waxy layer that prevents the loss of water, right? So at the very top, I would have my cuticle. After the cuticle, I would have my epidermis. Specifically, I would have my upper epidermis. And then, as you guys remember, we also used to have an epidermis on the bottom of the leaf. That was known as lower epidermis. Now, along with upper epidermis and the lower epidermis, so this is this area that you guys are seeing. This is the side of the leaf, okay? And then this area is the center of the leaf where the stem is running through. The stem is running from over here, okay? Like the vein of the leaf or the stem of the leaf is going through here. So this is the green part of the leaf. This is the green part of the leaf. And this is the center of the leaf. And the stem is coming through here. The stem is running from the middle of the leaf, okay? So what is the structure going to look like? First of all, let me tell you guys about the sides of the leaf, okay? Because the structure of sides of leaves, you guys probably remember quite well, okay? On the sides of the leaves, at the very underneath the upper epidermis, if you remember, we used to have a layer that was known as what? That was known as the palisade mesophyll layer, if you remember, okay? This used to be known as what? This used to be known as our palisade mesophyll cells, right? Then underneath the palisade mesophyll cells, we used to have another layer of cells, right? That another layer of cells actually included what? That another layer of cells used to be known as the spongy mesophyll cells, if you guys remember. Right? Now, this is the side of the leaf. So this was what we had on the side of the leaf. We used to have cuticle, upper epidermis, palisade mesophyll cells, spongy mesophyll cells, lower epidermis, and in the lower epidermis, we used to have our guard cells. Okay, if you guys remember, which were the opening of the stomata. Now, what about the center region? The region where the stem is actually present. What are we going to see? So in the very center, because we have the stem, right? So in the very center, we need to have the vascular bundle, okay? The vascular bundle is going to look somewhat like this. Over here in the very center, I'm going to have my vascular bundle. Okay. Now, in this vascular bundle, I am then going to have different parts. For example, I am going to have the outermost part is going to be known as xylem. Please remember that the labeling is different for different parts. In the roots, the outermost is phloem, but in the leaf, not in the stem. In the stem also, the outermost is phloem. But in the leaf, the center of the leaf, the vein of the leaf, I should not say stem of the leaf, the vein of the leaf actually has xylem on the inside. Then, on the inside of that, we will have our phloem. And then inside, underneath the phloem, we have another layer of cells. Now, what is this layer of cells called? This layer of cells has a very special name that you guys have not studied before. It is known as collenchyma. Let me write it out for you. And I will explain all of these cells to you in a bit as well. After we do the labeling, I will explain what these cells are. So we have xylem vessel at the very outside. Then we have phloem vessels in the middle. Then the outermost layer is a special type of cells that we call parenchyma. Parenchyma. Parenchyma are basically these sort of packing cells. Okay, they're the packing cells that actually help provide some support, some strength to the tissue. Now, after this, what's going to happen that the parenchyma cells are actually, when you're going to talk about it, the parenchyma cells are actually undergo a modification and they slowly modify into another type of cell. Okay. So I have my parenchyma cells. These parenchyma cells undergo certain modifications. I'll teach you guys what those modifications are. But these parenchyma cells can undergo certain modifications and they can go ahead and turn into a different type of cell. That new type of cell is going to be known as collenchyma. Collenchyma. Right? Collenchyma is, like I said, hold on, I'll correct the spelling. I think I wrote them incorrectly. Collenchyma are just like your parenchyma, but they have undergone a little bit of modification. So over here around the xylem vessel, you will see this parenchyma. And this parenchyma would, it would be like, you would slowly see that it would start to change and turn into collenchyma cells. So in this area, you would see like a transition. Okay, you would see a transition taking place. You would see that your parenchyma cells would slowly undergo transition and they would slowly modify and turn into collenchyma cells. Okay, now at the very top of this xylem vessel and at the very top of the phloem vessels, again we will have the collenchyma cells. So you will have collenchyma cells at the bottom as well as the top. And this altogether, your xylem vessel, your phloem vessel, and your parenchyma, if we combine these three layers, we get our vascular bundle. This is what our structure of leaf actually looks like, right? This is what the structure of leaf actually looks like. So this is our structure of leaf. Now, let me go ahead and show you guys a light microscope picture of this as well. Like if you guys see this under the microscope, what would it look like? Let me show you guys that. It looks like this. Okay. So this, the outermost layer that you see at the very top, this is the upper epidermis. Okay. Then these cells that you see over here, these are our collenchyma cells, which we will study about in just a bit. This over here, the tall cells are our columnar cells, palisade mesophyll cells. And these cells at the bottom are our spongy mesophyll cells. You guys can see the air spaces between the spongy mesophyll cells. Then these large cells on the outside are the xylem vessels. These small cells on the inside are our hold on, just give me 1 second. These cells that you see in the middle, the smaller cells, these are our phloem cells. Okay. And underneath the phloem cells, do you see this layer of cells? This layer of cells is our parenchyma. Okay. So this is over here, these large vessels that you see. These are xylem cells. Below the xylem cells, we have our phloem cells. Beneath the phloem cells, we have our parenchyma. These cells that you can see, these ones, this is all parenchyma. Then slowly see how the size of the cells is changing a bit. They're changing. They're slowly changing into a different type of cell, right? Which is the same as the cells in the top. The cells in the top and these cells at the bottom are similar. So this is the parenchyma cell, and see how they slowly change into bigger cells, into different cells. So the cells at the bottom are known as, again, your collenchyma. So these are the different types of cells that we are going to see in the structure of our leaf. Making sense? We understand the structure of our stem, the structure of our leaves, structure of roots. Does anybody have any questions about what we've discussed so far?
No.
Aruj, everything is making sense? No confusion? Okay. Now, I have told you guys a lot of these different structures, right? I have told you guys about what these structures are called. I've told you guys the labeling. Now, let's go ahead and actually study about these structures one by one. Let's start by talking about let's go over the structures of that we are going to see. So when we go ahead and we are going to talk about it, one of the structures that we are going to see is epidermis. I've mentioned epidermis again and again. In your leaf, in your stem, in your root, I told you guys the epidermis everywhere. What is epidermis? Epidermis is the continuous outermost layer of the plant. The outermost layer of the plant, right? That is known as epidermis. It is one cell thick, only one cell thick in size, right? And the major function, why do we need an epidermis? The reason we require an epidermis is because it is going to, is because it has a role in protection of the plant, right? How does it have a role in protection of the plant? So it has a role in multiple ways, okay? For example, like I just told you guys, in stems and leaves, it is covered by a waxy cuticle. In stems and leaves, it is covered by a waxy cuticle that prevents the loss of water, right? Then on top of that, it also has your stomata, which are surrounded by, or the opening is controlled by guard cells, if you guys remember, right? So your stomata allowed what? Which allowed transport of carbon dioxide in and out of the leaf, right? Then when we're going to go ahead and talk about it, in roots, the epidermis has extensions. What were the extensions called? Called root hair cells. Remember, we had studied them as well. They have got these extensions called root hair cells, which increase the surface area for absorption. So one of the major your component is the epidermis, and in leaves the upper epidermis is transparent. If you guys remember, you had studied it in IGCSE. Why was it transparent? To allow maximum light to reach the inner cells of the leaf. So this is what we had, remember, this is what we had studied about our topic of, if you guys remember, this is what we had already discussed about epidermis. Now, after when we move on from our epidermis, right? Okay, now what is the epidermis going to look like in a high power diagram? because we were talking about a low power diagram. What is the epidermis going to look like in a high power diagram? So in the high power diagram, the epidermis looks somewhat like this. You will see these will be your square cells like this. Now I'm drawing a high magnification drawing. Before this, I was drawing a low magnification drawing. This is a high magnification drawing. So you will have the cell wall, the cell membrane, the vacuoles, the nucleus, the chloroplast, all of that, right? Vacuole in the center. We will have our nucleus. You guys know the structure of plant cell. We will not be going over that because we have done the topic of cell biology, vacuole. All of this is going to be present. This is what? This is our epidermis. And on top of the epidermis, we can have, if you remember, I had told you that we can have a layer on the very top. This layer is going to be what? This layer is going to be your cuticle. So this is what an epidermis is going to look like in high magnification. Now, let's move on to the next structure that we have to discuss. What's the next structure that we have to discuss? The next structure that we have to discuss is our parenchyma. This was a new terminology that I've introduced to you guys today. So what is a parenchyma? Okay. So parenchyma, ENCHYMA. So your parenchyma, when you talk about it, it is sort of the supporting tissue of your plant. Supporting tissue, or a better word would be packing tissue. You know how when you guys, for example, transport something in cardboard boxes, so you put packing material around it, right? For example, you'll put the dishes in the cardboard box, but then you'll put a lot of newspaper or you'll put a lot of packing material all around it to provide some strength and support. That's parenchyma. It's the packing material of the cell, of the plants. So parenchyma, what are they? They are thin walled cells. They're really thin walled cells used as packing tissue. So when they want to pack the cells together to provide support and strength, parenchyma is the tissue that they go for, right? The cells are metabolically, when we talk about them, active, right? They're metabolically active, meaning that they will be involved in metabolism of the plant, such as you can use them for storage of starch. Like if I want to store starch, so I can store them in the parenchyma cells, right? When they are fully turgid, they will provide a lot of support to the plant. So when the plant is cells are going to be fully turgid, fully filled up with the water, fully swollen up, they will provide a lot of support, right? And they prevent wilting because they provide support, so they don't let the plant wilt. Then they have air spaces, which are really important because they allow the gas exchange to take place. And when we're going to talk about it, water and your minerals are also through the transported through the cells. And we will study this. I will teach you guys the pathway that we have got two pathway, aplastic, symplastic pathway that we have to do. It's in our topic. So the pathway that water will take and how they will go through the parenchyma tissue, we will go ahead and take a look at that as well. Now, parenchyma, the cortex, remember I taught you guys about the cortex in root and stems. If you guys remember, the cortex in root and stem is made of parenchyma. That's what the root and cortex is actually made up of. They are made up of parenchymal cells, right? They the parenchymal cells, these cells also contain chloroplast, by the way. So they can be modified. They can be, modified to make the palisade cells and your spongy mesophyll cells as well. So they can undergo modification. I can take my parenchymal cells, I can modify them, and I can make spongy mesophyll cells or palisade mesophyll cells. Why can I do that? Because they do have what do they have? They have my chloroplast present inside of them. What do the structures cells actually look like? So the cells actually look something like this. So it will literally look like, like you've taken a bunch of tissue paper and you've sort of stuffed it all together. Like it literally, it's the entire purpose is that it's supposed to be, packing tissue. So that's what it looks like a bunch of cells that have been thrown together. That's literally what it looks like. Like you've taken a bunch of cells and you've stuffed them together. And when you're going to talk about the structure of the cell, so the structure of the cell is going to be quite similar to what you guys already know. You're going to have your cell wall, you're going to have your cell membrane, you're going to have your vacuole, you're going to have your chloroplast, right? You're going to have your cytoplasm. So all of these structures are pretty much going to be the same. So this is what your parenchyma is, right? This is the vacuole in the center. This is our. This is going to be our cell wall. This is our cell membrane. If you guys remember the structure of the plants, this is our cytoplasm. I've drawn a lot of cytoplasm. Normally, they don't have this much. This will be our nucleus. And this, of course, in the center is going to be our vacuole. So this is what your structure of parenchyma is actually going to look like, right? And then I can take this same parenchyma and I can modify it. And I can modify it and I will get collenchyma. That's it. Collenchyma is nothing different compared to parenchyma. There is a small difference when we talk about parenchyma and collenchyma. The small difference is this, that in collenchyma, I have taken the same cells, I've taken the same parenchyma cells, and the same parenchyma cells, I have gone ahead and I have added extra cellulose around the corner of the cells. That's it. That's the modification, right? They are modified parenchymal cells that have extra cellulose around the corners of the cell, right? They have the around the corners of the cell. This provides extra strengthening. It provides extra strengthening and support because that cellulose makes the cell wall stronger. So it provides even more strength. It provides even more support. And when we're talking about remember the center of the leaf over here? This center of the leaf, by the way, we call it midrib. Okay, this center region is known as midrib. midrib, midrib. So the midrib, that's why it had a lot of collenchyma, because the extra cellulose actually provides a lot of strength and support. Then another important structure is that we have the endodermis. Now, I'm not going to tell you guys the role of endodermis right now, because when I teach you guys the roots in detail, like the function of roots and how the roots work, then we will discuss the role of endodermis. But endodermis is similar to epidermis, meaning that it is one cell thick and it surrounds what? It surrounds the vascular tissue in stem and roots, right? Then we have our final important structure is pericycle. I mentioned pericycle to you guys in the structure of roots. Now we can understand what pericycle actually is. Pericycle is a layer of cells. It's literally a layer of cells, and it's just inside the endodermis. Like wherever your endodermis is, it's normally just inside the endodermis and next to the vascular bundle. So it normally surrounds the vascular bundle. That's what you can say. It surrounds the vascular bundle, right? In root, it is one cell thick. In roots, it's just one layer of cells, and it can um, grow new roots. The pericycle can actually divide and grow new roots. In stems, where do we find it in stems? Because I did not tell you guys the labeling of pericycle in stems. In stems, it is formed from a tissue called sclerenchyma. What is sclerenchyma? I will teach you guys. We will go over what sclerenchyma is. But sclerenchyma, remember those fibers? I told you guys that we have these fibers, and I'll tell you guys what the fibers are. There's sclerenchyma fibers, right? So the pericycle is the layer of cells that we see around the vascular bundle in the root. And the pericycle is composed of these fibers in the stem. In the stem, the fibers are the ones that are making the pericycle. Okay. In the root, the pericycle is just a layer of cells that is present all around the vascular bundle. And we will study what sclerenchyma is. Okay. We will study this in detail. But just to give you an idea, what is your sclerenchyma? Sclerenchyma are sort of like xylem vessels. Okay. These are dead lignified cells, like they have a lot of lignin, if you guys remember what lignin is, for extra strength, right? So now, so making sense? Anybody has any question? Would anybody like to ask anything? Anybody has any confusion? Khadija, Aruj, is everything clear? I hope no one has any confusion. No questions, right?
No.
Okay. Okay, guys, then we're going to go ahead and we're going to stop it over here for today. And inshallah, from the next class, now we will take a look into detail in the structure of our xylem vessels, phloem vessels, and how the transport actually takes place. Okay? Okay, guys, thank you so much. I'll see you in the next class. Have a good day.